Welcome back, everyone. Today we're going deep, really deep, into the world of ligand-gated ion channels. You know those tiny little gateways on our cells?
Ah, yes, LGICs. Quite fascinating, aren't they?
They really are. Controlling everything from muscle movement to how our brains work, it's incredible. Almost like those sci-fi force fields, you know?
Yeah.
Need a specific code to open them.
Haha. Love that analogy. It is like a highly selective doorman, isn't it? Only allowing certain molecules to trigger the opening. And in a busy place like a cell, things need to move fast. That's where these ligand-gated ion channels really shine.
So we're talking about speed, but also security. Now, you mentioned these LGICs. They're different from voltage-gated channels. What's the key difference there?
Okay, well, think about it this way. Voltage-gated channels, they're triggered by changes in the electrical charge, right? Across the cell membrane, like an automatic door, senses a change and whoosh, it opens.
I get it.
But with LGICs, it's different. They need a specific molecule to bind to them, and we call those molecules ligands. It's a much more targeted system, almost like a lock and key.
So not just any molecule can open these LGICs then?
Exactly. Specificity is key here. This ensures that only the right signals get through, and at the right time, too. Imagine the chaos if any molecule could just barge in.
Total mayhem. So tell me, what kinds of molecules are we talking about here? What are these keys in this cellular world?
Well, it really depends on what type of LGIC we're looking at, but some of the most famous ones are neurotransmitters.
Neurotransmitters, right. Those are the chemical messengers in the brain, right?
That's right. They're how neurons talk to each other. So imagine one neuron wants to send a signal. It releases neurotransmitters, could be serotonin, dopamine, any number of them.
Okay.
And these neurotransmitters, they act as the key to unlock a specific LGIC on the receiving neuron.
I see. So when a neurotransmitter, let's say serotonin, binds to its specific LGIC, then what happens?
Well, then the channel undergoes what we call a conformational change. It basically shifts its shape, opens the gate to allow ions to flow in or out.
Ions. Okay. What's their role in all of this?
Ions, they're just atoms with an electrical charge. And their movement across the cell membrane, that's what creates electrical signals. So when the LGIC opens up, it allows certain ions to flow, and that changes the electrical charge of the neuron.
Like flipping a switch.
Exactly. That's what ultimately causes the neuron to fire a signal down its axon to communicate with other neurons. And the amazing part is just how quickly this all happens.
Let's talk speed then. Why is it so important for these channels to open and close so fast?
Well, think about something like reflexes. You touch a hot stove, you pull your hand away instantly, right?
Of course.
That kind of lightning-fast response just wouldn't be possible without the super-fast signaling that these LGICs allow. Imagine if your brain took, seconds to process every word you hear.
I can't even imagine.
Exactly. It's all about speed, not just for reflexes, but for our thoughts, our reactions to the world around us. And part of how they achieve this speed is because the ligand doesn't have to stick around. It triggers the channel to open, and then, poof, it detaches.
It's like a rapid-fire system. So efficient. Now, what about the channels themselves? What are they actually made of?
Well, they're typically made up of several protein subunits, sort of like Lego bricks. Each one comes together to form this larger structure. And these subunits, they create a pore through the cell membrane, like a selective passageway for ions.
I bet the arrangement of those subunits is super important, right?
Yeah.
It probably determines what kind of ligand can bind, like a key fitting into a specific lock.
Spot on. It's like a custom-made binding site just for its corresponding ligand. It's another layer of security, making sure only the right key can open the door.
Okay, so we have these cellular doorways, tightly controlled by specific ligands, capable of lightning-fast responses. But where are they? Just scattered all over the cell? Or do they tend to congregate in specific areas?
Well, in the nervous system at least, they're often found clustered together at the synapses.
Synapses. Remind me, those are the tiny gaps between neurons where signals are transmitted, right?
You got it. And by clustering at these communication hubs, it ensures that signal transmission is both efficient and rapid. And remember, speed is essential for the nervous system.
Right, makes sense. Now, you mentioned that these channels, they're not just in humans, right? We see them in other species too.
Absolutely. The world of LGICs is incredibly diverse. We see them in everything, from bacteria to plants to animals. And the specific ligands involved, they're tailored to each organism's needs.
So even plants have these sophisticated signaling systems, but with their own set of keys.
It's a testament to just how adaptable life is, wouldn't you say? And it really makes you wonder about how these channels evolved Why is it beneficial to have such a diverse array of LGICs with specific ligands?
That is a great question. Why would our bodies evolve such a complex system? What's the advantage of having all these specialized channels?
Well, scientists are still exploring that, but one possibility is that it allows for a much finer level of control over all sorts of cellular processes. Instead of one generic channel that responds to any signal, you have a whole array of specialized ones, each one perfectly tuned to a specific ligand.
Like having a toolbox full of specialized tools, each one perfect for a specific job.
Exactly. And that specialization is essential for how our bodies function. It's complex, but is also incredibly nuanced.
Makes me think about the potential for drugs that target specific LGICs. If we could really understand this lock and key mechanism for each channel.
We could potentially design drugs that either mimic the natural ligand, what we call agonists, or block it completely. Those are antagonists.
Wow. It's like we could unlock new ways to treat all kinds of diseases and disorders.
And that's why research into LGICs is so exciting. It's not just about understanding basic biology, but also about unlocking the potential for new and innovative treatments.
So we've covered a lot of ground today, from the basic workings of LGICs to their role in our health, and even touched upon the potential for developing new therapies.
And before we wrap up this first part of our deep dive, I want to leave you with something to think about. We've talked about how diverse LGICs are across different species, but even within a single organism, why might it be advantageous to have multiple subtypes of LGICs that respond to the same ligand?
That's an interesting question. What are the implications of having this redundancy in our signaling systems?
It's certainly something to ponder. It's a bit like having backup generators.
Yeah.
For your cells. If one type of LGIC isn't working properly, another one can maybe step in and keep things going.
Oh, okay. Like a safety net for cellular communication. But let's go back to this lock and key idea for a second. You mentioned earlier that each LGIC has this special binding site for its ligand. Can you give us a bit more detail on what that actually looks like? On a molecular level, I mean.
Sure. Imagine a tiny pocket on the surface of the channel. That's the binding site. It has a very specific shape, very specific chemical properties too. And the ligand, well, it has its own unique shape and chemical makeup, and it has to fit perfectly into that pocket, like a hand fitting into a glove, I guess you could say.
So it's not just about the shape, it's about how the chemicals in the ligand and the binding site interact with each other too.
Right. There are these specific amino acids. Those are the building blocks of proteins, and they interact through different forces like attraction and repulsion. It's like a very specific chemical handshake, and it helps stabilize the binding, make sure that only the right ligand can fit in.
It's all so precise, it's amazing. You mentioned before that there are all sorts of LGICs in different species. Do any particularly unique ones come to mind?
Well, one that I always find fascinating is the melanocortin receptor. It's found in a whole bunch of animals, from fish to mammals, and it's involved in some really diverse functions.
Oh, like what?
Well, things like regulating pigmentation, how much color an animal has. And then it also plays a role in controlling appetite and even influencing sexual behavior.
Wow, that's quite a range. It's incredible that one receptor can be involved in so many different things.
It really shows how versatile these LGICs are. Evolution has really put them to work in all sorts of ways.
Let's go back to speed for a minute. You said that these channels can open and close in milliseconds. But how is that even possible? I mean, it seems like it would take much longer for a ligand to bind and then for the channel to actually open.
Well, remember how we talked about those LGIC clusters at synapses? The neurotransmitters, those are the ligands in this case, they're released really close to the LGICs. It's not like they have to travel a long distance.
So being close to their target makes things faster.
Exactly. They don't have to travel far, so it doesn't take as long for them to bind and trigger the channel opening.
And once the ligand is bound, those conformational changes in the channel, those happen quickly too.
Oh, yeah. Those are really fast. It's like, think of it like a machine, different parts moving together. It's very precise. And those movements happen incredibly fast.
So it's that combination then, the ligands being close by and the channel changing shape so quickly. That's what allows LGICs to be so efficient.
That's right. And remember, the ligand doesn't have to stay bound for long. Once it's triggered the channel, it can detach. That allows the channel to close quickly and get ready for the next signal. It's this constant cycling on and off that allows for really precise control over the ions flowing in and out.
We've talked a lot about LGICs in the brain, but they're in other parts of the body too, right? Can you give us an example of how they work outside the nervous system?
Sure. Muscle contraction is a good example. LGICs play a key role at the neuromuscular junction. That's where a motor neuron connects to a muscle fiber.
Okay, so how does that work? How do the LGICs fit in there?
Well, the motor neuron releases acetylcholine, which is a neurotransmitter, and it binds to LGICs on the muscle fiber. Specifically, these are called nicotinic acetylcholine receptors.
Okay.
And that binding triggers a cascade of events that ultimately leads to the muscle fiber contracting.
So every time we move a muscle, LGICs are involved.
You got it. And again, that speed, that precision of the LGICs, that's crucial for coordinated movement.
Earlier, you mentioned something about LGICs having different numbers of subunits. What determines how many subunits a particular channel needs?
It really depends on the channel and what it does. Some might only have a few subunits, others might have 5 or even more.
Why is there so much variation?
It's sort of like building with Legos again. You can create different structures depending on how many bricks you use and how you arrange them. The same idea applies to LGICs. The number of subunits can change its properties.
Properties like?
Well, things like how sensitive it is to ligands, or how fast it opens and closes, or what kinds of ions it lets through.
It's like another level of fine-tuning then. It makes them really adaptable.
Exactly. That adaptability is one of the things that makes LGICs so important in so many different biological processes.
I'm curious about how they're regulated. We've talked about how ligands bind and trigger channel opening. But what other factors can influence their activity?
Oh, there are a lot of regulatory mechanisms, both inside the cell and from the outside environment. It's like a control panel with lots of different dials and switches.
Can you give us some examples?
Sure. Phosphorylation is a common one. That's when a phosphate group is added to the channel protein. It can either boost its activity or inhibit it. It's like a dimmer switch for the channel.
So there's more control than just on or off.
Absolutely. And another mechanism is trafficking. Remember how LGICs cluster at synapses? Well, the cell can control how many channels are there by moving them around to and from intracellular compartments.
So it's like there's a reserve of channels waiting to be used.
That's a good way to think about it. And all sorts of things can affect trafficking. Neuronal activity, what stage of development the cell is in, even drugs.
This is getting really complex. There are so many ways to control LGIC activity.
That complexity allows for the incredible precision and adaptability of our nervous systems.
Now let's talk about what can go wrong. You mentioned that there are diseases and disorders linked to problems with LGICs. What kinds of problems can happen?
Well, one common issue is mutations. Changes in the genes that code for the LGICs. Those mutations can change how the channel is structured, and that can lead to all sorts of problems.
Like what?
Well, for example, there's a condition called myasthenia gravis. It's caused by mutations in the gene for the nicotinic acetylcholine receptor. And it causes muscle weakness and fatigue because the signaling between the nerves and muscles is messed up.
And I imagine that mutations in LGICs in the brain could lead to neurological problems.
You're absolutely right. Mutations in the LGICs that bind to GABA and glutamate, those have been linked to things like epilepsy, anxiety disorders, even schizophrenia.
So these tiny channels can really have a big impact.
They really can. That's why research in this area is so important. By understanding how they work and what can go wrong, we can hopefully develop better treatments for all sorts of conditions.
I'm also thinking about toxins. I know some venoms and poisons can interfere with LGICs. How does that work?
Yeah, some toxins can overstimulate the channels. They act like agonists. Others block them completely, acting as antagonists. It's like they're hijacking the channels, and that can have some pretty serious consequences.
Can you give us an example?
Sure. Think of the black widow spider. Its venom has a toxin that targets calcium channels at the neuromuscular junction. It causes a huge release of acetylcholine.
And what does that do?
It causes really bad muscle spasms and paralysis. It's a good example of how messing with LGICs can be really dangerous.
That sounds scary. It really makes you appreciate how important that balance is.
Definitely. And it shows why it's so important to understand these channels and how toxins affect them. That knowledge is key for developing treatments for different types of poisoning.
Let's switch gears and talk about drug development. You said before that developing drugs that target LGICs is a promising area. What are some of the challenges and opportunities there?
One of the biggest challenges is specificity. Remember, there are lots of different LGICs, and they share some similar features. So it can be tough to design a drug that targets only one type of LGIC without affecting others.
So it's like trying to make a key that fits only one lock, when there are lots of similar-looking locks around.
Exactly. And if a drug binds to multiple LGIC types, it can cause a lot of side effects. It's like throwing a wrench into several cellular processes at once.
I can see how that would be a problem. But what about the opportunities? What are some of the potential benefits of targeting LGICs with drugs?
Oh, the potential is huge. Because LGICs are involved in so many vital processes, drugs that can target them could be used to treat a wide range of diseases. We're talking about fine-tuning the very signals that control our bodies and minds.
Can you give us some specific examples?
Sure. We already have drugs that enhance the activity of GABA receptors. Those are involved in inhibiting neuronal activity. They're used to treat anxiety disorders and epilepsy. And then there are drugs that block NMDA receptors. Those are involved in learning and memory, but they can also contribute to neuronal damage. So they're being looked at as potential treatments for Alzheimer's disease and stroke.
So potentially groundbreaking treatments for some serious conditions.
That's right. And as we learn more about specific LGIC subtypes, we'll be able to develop even better treatments, ones that are more targeted and have fewer side effects.
This is really exciting. It feels like we're at the beginning of a new era of drug development, one that's based on understanding those molecular machines inside our cells.
And LGICs are leading the way. They may be tiny, but they have a lot of potential.
We've covered a lot of ground in this episode, from the basics of LGICs to their roles in health and disease and the possibilities for new treatments.
It's been a fascinating look at these amazing channels.
And as we wrap up part 2 of our deep dive, I have one last question. We've talked about how LGICs cluster together at synapses, but how do those clusters form in the first place? Is it random, or is there a specific process?
That's a great question, and it leads us nicely into the final part of our discussion. It turns out that those LGIC clusters don't just appear randomly. It's a very organized process involving a complex interplay of different molecules and mechanisms.
So there's a system in place. It's not just LGICs hanging out at the synapse by chance.
Right. It's like, it's like a carefully planned dance. There are these molecules that make sure everything ends up in the right spot.
A synaptic dance. Okay, I like that. Tell me more about who the dancers are in this scenario.
One of the most important groups is the scaffolding proteins. They're kind of like the organizers, they provide support and make sure the LGICs go where they're supposed to.
So they're like the stage crew getting everything set up for the big show.
Exactly. They create the framework, the structure that holds everything together. It's all about making that signal transmission as smooth as possible.
And are there other molecules involved in this organization?
Oh, yeah. Cell adhesion molecules are important too. They act like bridges connecting the neuron that's sending the signal to the neuron that's receiving it.
So they're making sure the signal has a clear path.
Right. And those adhesion molecules, they help position the LGICs right at the point of contact between the neurons. That way, they're in the perfect spot to grab those neurotransmitters as they come in.
This is all very intricate, very coordinated.
It is. And what's really cool is that it's not static. The clusters of LGICs, they can actually change, depending on what's happening at the synapse.
So it's dynamic. It can adapt.
Exactly. The synapse is constantly responding to changes in activity. Connections can get stronger or weaker depending on what's going on. And that plasticity, that's what allows our brains to learn to adapt to new things.
That makes me wonder about learning and memory. If those LGIC clusters are changing based on activity, could that be part of how memories are formed? By strengthening or weakening connections between neurons.
You're thinking along the right lines. That's one of the big ideas in neuroscience right now. The thought is that those changes in synaptic strength, which are influenced by those LGIC clusters, well, they could be how we learn and remember things.
So these tiny channels, they could be the key to understanding how our brains store information and adapt to the world around us.
It's pretty incredible, isn't it? These tiny channels working away in our brains, shaping our thoughts, our emotions, our memories.
It really makes you appreciate just how complex the brain is. We've learned so much in this deep dive, from how LGICs work at the most basic level to all the different roles they play in our bodies, and even how they might affect learning and memory.
It's been a fascinating journey. These channels may seem simple at first, but they're anything but. They're like tiny, intricate machines that have been shaped by evolution to control all these essential signals that make life possible.
Thank you for joining us on this deep dive into the world of ligand-gated ion channels. We hope you've gained a new appreciation for these tiny gateways that play such a big role in our lives. And until next time, keep exploring and stay curious.